Okay, so this video is to explain to you your mini essay task. Okay, so some of you will know and some of you this will be completely new to, and this is the first time and it's just to get us started. That you will know, hopefully most of you, that at the end of paper 3 you will have to write a biology essay. It's only going to take about 45 minutes in that paper, but you will need to try and make it count because it's worth 25 marks on that paper. So it's a big chunk of marks. And you'll be asked to write a biology essay on a big topic idea. So it's not going to be a really small idea, okay? It's going to be a really big idea. So previous topics have been cycles in biology, discuss. The importance of ions in biology, discuss. DNA and its importance as an information carrying molecule, discuss. So they're not little tiny topics, they're big, big ideas. And really what the examiner is trying to do is to test how well you can think about the big picture of that idea and link it to other areas of the course that you've studied. Okay, so you will need to explore the importance of biology, that particular topic, like in the case of ions, to the whole course. So you'd link it to different topics. So for example, with the ions essay, you probably remember meeting sodium ions in co-transport. So that's one place in the course where you can talk about the importance of one ion type. We could also talk about the importance of mineral ions moving in by active transport to the base of your xylem, okay, as a bit of root pressure, pushing the water up the xylem vessels. We could also talk about the importance of our ions within our hemoglobin, our H plus ions in our Bohr-Schiff, etc, etc. So we had a lot of ions last week. So that's what we'll be trying to do, make a lot of links from the big topic area of the title to as many different topics as possible. Now that's our aim eventually, but for this mini task I'm not going to ask you to do that. I'm going to ask you to just think about one, okay? But to help us start practicing this skill, I just want to show you the big mark scheme so you can have a look at this. Now we're going to be aiming for the top, obviously, between 21 and 25 marks. To get this, you can see it's pretty tricky, but the thing I want us to focus on is to get into this boundary, to get above 20. We need to make sure that our response links several topics to the question. Okay, so that's what we're going to focus on, linking a topic to the main theme. Okay, so we're going to pick a topic, hopefully, for you to develop and link to a question as a starting point. Please, by all means, have a look through. There are other things we'll focus on at different points. For some of you, you'll read this kind of at the top end here. To get top marks, you need to read beyond the specification. In year 2, I do special workshops where we cover extra material outside the spec for that, and I'll tell you about that at the beginning of next year. So at the moment we're just starting this tiny bit of development in practicing this skill. So exploring the importance of this biology topic to the whole course. We're going to pick a place in the course where you can show it's really important. Okay, so I have prepared for you 3 extracts for an essay title that's already been, and the essay title that we're not preparing for, this is just an example, is DNA and its importance as an information transfer molecule in biology. So that was the title, and the students had to pick maybe 4 or 5 different places where they had met DNA transferring information in biology. Okay, so they may have picked during DNA replication and then mitosis, meiosis, cell division. So where the DNA is copied and then passed as an information transfer molecule to the 2 daughter cells. They might have picked DNA from a virus infecting a cell, transferring the DNA for how to make a virus to the cell, and to discuss the importance in that process. They might have talked about DNA as you would, hopefully, by the time you get into year 2 in genetic engineering, so cutting a gene out of one organism and putting it into another and giving that organism a new feature. Okay, so again, DNA working as a information transfer molecule. And they also may have talked about transfer to other humans through gene therapy, or to crop plants to increase their yield or make them heat resistant. Anyway, lots of different examples. So the idea is that they would have picked different areas of the course where they've met DNA as a transfer molecule, and then made sure they develop why that's so important in biology, that particular role of DNA as a transfer molecule. So I've given you 3 different student extracts for this title where they're just doing one role of DNA, and it's the same role. So this is student 1. DNA is a double helix found in the nucleus of all cells, which stores all the genetic information. When the cell is going to divide, the DNA unzips and 2 strands are copied identically, producing 2 new daughter molecules. This allows the genetic information in the DNA stored in the code ATGC to be passed on to each new cell, so they have all the information they need to function. Okay, I want us to just reflect on that for a moment. So they have talked about DNA as a transfer, genetic information transfer molecule. So there's an information transfer implied in this. But look at the material they've presented. Is any of it A level? Okay, keep it in front of you. You've got the Word document. My feedback to this student was this: you've written at GCSE level. There's nothing there you didn't know when you joined me at the start of year 1. There's no detail that has been taught to you in the A level course. The other thing is, although they have described how it's an information transfer molecule, it's not said why this is important, other than so that they all have the information they need to function. So it's beyond vague. So they've not addressed the title, the importance of this DNA acting as an information transfer molecule in this case. And they've also not linked to any other topics. So it's basically a grade D extract. Okay, have a look at the next extract. DNA is the genetic information storage molecule found in all cells. It is a linear molecule within eukaryotic cells and circular molecule within prokaryotic cells. So there's a bit of A level in there, but has the same function in both: to carry the genetic information for the primary structure of all the proteins a cell can make. When a cell divides by mitosis or meiosis in eukaryotic cells, or by binary fission in prokaryotic cells, it is important that it is an accurate copy of the DNA, is made so that vital information can be passed to the new daughter cell. This is done by DNA replication. The DNA helicases break the hydrogen bonds and unzip the double-stranded DNA, exposing the bases. DNA nucleotides bind to the exposed bases by complementary base pairing, guanine to cytosine and adenine to thymine. DNA polymerase joins adjacent DNA nucleotides together by condensation reaction, forming phosphodiester bonds. The two molecules are identical and the process is essential to allow DNA to transfer information for how to make proteins to each daughter cell. Okay, so feedback, what do you think? Can you see keywords and detail you have learnt in the A-level course? Yes. But when you get to the importance here, the two molecules are identical and this process is essential to allow DNA transfer information for how to make proteins to each daughter cell. They just missed the boat here. All they needed to do is to say, and without it, for example, name a protein and say why a cell wouldn't be able to function without it. So you might say, for example, if this was a salivary gland cell and it wasn't able to make amylase, you wouldn't be able to break down starch. And if you couldn't break down starch, you would then not be able to break it down into maltose for maltase, and there won't be glucose, won't be a respiratory substrate. So you can see they've not really developed the importance on there. Okay, now remember, this is kind of the level you'd be writing at year 2, at the end of year 2 as well. So good level of detail, just haven't developed the importance yet. So better biology, that's kind of what I want to see, lots of key terms, biological names, links to other topics if you can. So you're not just talking about one area of biology you've been taught in one topic, but you're making links between different bits. But they didn't really explore the importance of transferring that DNA information to the daughter cell, so it lacked detail. So you can't really get past 11 out of 12 in that mark scheme. Okay, so I'm pretty sure you'll get this from what I've done now. So if we were looking at a very good answer, there'd be something you would definitely see. So it's the same area of DNA working as a information transfer molecule. But this student, you can see in this example, has got not just one paragraph, but 2 paragraphs. And the first paragraph I call the role paragraph. So here they outline the role of DNA as an information transfer molecule from parent cell to daughter cell. Okay, so again, good quality, lots of key terminology. Each gene found in DNA has a base sequence which codes for the amino acid sequence of a polypeptide. Okay, we also know that includes functional RNA now, but this is okay. It doesn't have to be perfection, just biologically correct. So this information is essential for protein synthesis, so correct cellular function. So when the cell divides by mitosis, each daughter cell must receive an accurate copy of the base sequence information. This is achieved most of the time due to the semi-conservative nature of DNA replication. During the S phase of the cell cycle, prior to cell division, DNA helicases break the hydrogen bonds, unzip the DNA, the double-stranded DNA. Free DNA nucleotides bind to exposed bases. By complementary base pairing, guanine to cytosine, adenine to thymine. DNA polymerase joins adjacent DNA nucleotides together by condensation reaction, forming phosphodiester bonds. The 2 DNA molecules are identical. They condense around histones to form a chromosome made up of 2 sister chromatids joined at the centromere in prophase of mitosis. The chromosomes then line up on the equator of the spindle in metaphase. Then the sister chromatids are separated and pulled to opposite poles of the cell by shortening of the spindle fibres in anaphase. Finally, a new nuclear membrane forms around the chromosome and the cells divide in two, producing two daughter cells with a full set of genetic information which has been transferred accurately. So here they've outlined the role of DNA as genetic information for coding for polypeptides and talked about how it is transferred accurately to daughter cells. Okay, but they haven't yet said why that's so important. Now, that was the role paragraph. Now, it is a very comprehensive paragraph and nothing near what I would expect right now from a student. But this is the kind of detail that we want, lots of key terminology, linking topics together in that role paragraph as much as we can. But what we need to do is to make sure we follow that with an importance paragraph. So you then do a short paragraph after your role paragraph outlining the importance. Okay, so the importance of this transfer of genetic information by the DNA during this process is illustrated by when it goes wrong. This can happen in two ways. You can get a base mutation or a chromosome mutation. Now I need to say here that we're going to talk about base mutations next week. Chromosome mutations we won't study till year 2. Okay, so a base mutation occurs during DNA replication when the DNA nucleotides align on the complementary bases of both strands. Sometimes the wrong DNA nucleotide lines up, you get a substitution, or it's added, or is missed, producing a new version of the gene, a new allele, with an altered base sequence. This can result in the cell having different information, which can result in a non-functioning protein. Now, as this is a year 2 student, they're just doing their final exam, they're able to draw on year 2 knowledge here as well. So they can talk about a non-functional protein, which is a proto-oncogene mutation causing the production of an oncogene. An oncogene is a gene that causes cancer. Okay, so they're able to make a link here to if this transfer went wrong in this way, the importance would be the protein synthesized when this allele is expressed by the cell will cause overstimulation of cell division, resulting in uncontrolled mitosis and tumour formation, resulting in cancer. Okay, so at the end of year 2, when we've got all that knowledge and we've revised all of our year 1, within this essay we would expect some role paragraphs like this with lots of different detail and links to other topics, followed by an importance paragraph. Okay, and for the total essay they'd maybe do that 4 times. Again, something we will practice in year 2 as well. Okay, so what do I want us to do this week? Nothing quite like this yet. This is just to see where we're going. I'm going to give us a bit of feedback now on that paragraph. So it's really because the student used 2 paragraphs here. In the first paragraph about the role of DNA as a transfer, information transfer molecule from parent cell to daughter cell, the A-level terminology was very strong. There were many links to topics in there as well. But also by structuring a role paragraph followed by an importance paragraph, it kind of already forces you to do that importance properly. And as soon as you start talking about the importance and linking back to the title, you skip up past 11 and start working your way up that mark scheme, which is where we want to start to progress. So your mini task. Okay, your mini task is going to be about a protein. Okay, so you're going to write and submit to your biology tutor just one role and one importance paragraph for the title, proteins and their importance within living organisms. You need to pick a protein that you've met in the course and be happy you can describe its role and then write a second paragraph about why you think this role is important. The best way to do this is to imagine what would happen what would stop in that organism, in that cell, in that system, if that protein wasn't there. Okay, so I'm going to let you check out this example, but pick your own. So I've gone for reverse transcriptase. I thought there's no way any of you'll pick this one. And I said, what's its role? So we met as an enzyme found in HIV. So I said it's found in HIV. HIV's got a simple structure. It's got its genetic material, which is RNA and reverse transcriptase enzymes. They're surrounded by a capsid, a protein coat, which in turn is surrounded by an envelope containing attachment proteins. When HIV binds to the T cells by its attachment proteins, it passes into the cell via the phospholipid bilayer. Inside the cell, it breaks down and it uses its RNA and that reverse transcriptase to make more viral RNA and reverse transcriptase and capsid proteins. The reverse transcriptase actually joins the host's own DNA nucleotides together by condensation reactions to produce DNA copies of the viral RNA. These are inserted into the host DNA and the host cell then produces viral proteins via transcription and translation, you could say via protein synthesis. The new viruses assemble, and as they bud out of the cell, they take some of the host cell membrane with them as a new envelope. So that's the role of reverse transcriptase within HIV infecting a cell. So then once I've done my role paragraph on how the enzyme works, what it does, and you can see it's just like a description, like a 5 marker, and then follow it up with an importance paragraph. So I say reverse transcriptase is therefore a very important enzyme to HIV. Okay. So I said to you, why, if you're trying to do this importance paragraph, you have to think about what would happen if it didn't have it. Okay, so as the host human T cell, it in fact doesn't have the enzyme. So human cells don't have reverse transcriptase because human cells have DNA as their genetic material, so they don't need it. Okay, they don't need to convert RNA base sequence into DNA. So if the HIV did not insert the enzyme, so if it didn't have the enzyme, didn't insert it along with its viral RNA, it would not be able to infect the cell, as the DNA copies of the viral genetic information could not be made. No new viral RNA or proteins needed to make new virus would be produced. So without reverse transcriptase, HIV cannot infect human cells. Okay, so you can get into them, but it can't cause those cells to make new viruses. Now, that's all I want you to do. Okay, a role paragraph and an importance paragraph. Pick one protein you've met in the course so far, okay, where you can quite nicely talk about its role. Maybe it could be the co-transport protein for co-transport of glucose, or it could be the endopeptidase is involved in protein digestion. Okay, you can pick any named protein. Happy you can write a nice paragraph about its role. Then you need to imagine that that protein doesn't exist and what the impact would be for your importance paragraph, and it can be a nice little short paragraph there. Okay, and that's what I would like you to submit to your tutor. Now, if you want a super challenge, okay, it may be that in your importance you might be able to develop it on a little bit. So as I was writing this, I remembered that some of the drugs, the antiviral drugs that you treat HIV with, actually are inhibitors of reverse transcriptase. So I've kind of been able to illustrate that. I looked up the name of one of these drugs and I said the importance, this importance that I've described of the enzyme is shown by the action of these inhibitors, these viral drugs that inhibit reverse transcriptase, because taking the drug stops new viruses from being produced by stopping reverse transcriptase from working. So it kind of illustrates what happens when that enzyme doesn't work for the virus. And I thought it was a nice little bit of getting in outside spec content, which is what we collect along the way in year 2. Okay, so have a go. Simply any enzyme, any protein, it could be antibodies. So you could talk about antibodies and their importance, what they do, and then what would happen if you didn't have them. Okay. Or it could be, as I said, the co-transport protein for glucose, how it works for a paragraph, and then imagine what would happen if you didn't have it, so why it's important. And that's what we would like you to submit. Okay.